Okay, dear students, in the previous class, we were discussing about codominance. Example is a ABO blood group that is controlled by I gene. ABO blood group is controlled by I gene. ABO blood group controlled by I gene. ABO blood group is controlled by I gene. The gene that controls the production of sugar polymer antigen, I think I explained this. And the gene I has the following alleles. Gene I has following alleles. They are IA, IB and IO. So the three alleles are IA, IB and IO. IA, IB and IB have sugar polymer on their surface, but IO is not having sugar polymer on their surface. Sugar polymer on their surface. Now, let us make a chart. In that chart, alleles from parent 1, alleles from parent 1, from alleles from parent 2, then genotype of offsprings, blood group phenotype. Blood group phenotype. If father gene is, parent 1 gene is IA and parent 2 second gene is IA, then the genotype of offspring will be IA and the blood group is A. Blood group is A. Then alleles from parent 1 is IA and parent 2 is IB, and the blood group will be IA, IB. That means AB will be the phenotype. If parent is IA and the second parent is IO, the genotype will be IAIO, blood group is A. If one parent is IB, second parent is IA, then the allele will be IB, IA, blood group is AB. Then if the parent blood group, one parent is IB, another parent is IO, IBIO, blood group is B. If one parent is IO, another parent is IO, blood group is O. So this is the chart which shows the different blood group phenotypes. When IA and IB are together, when IA and IB are together, both they express, both together they express their own sugar and maintain their identity. Maintain their identity. They maintain their identity. This is called codominance. This phenomenon is called codominance. See, IA, IB, they come together. Even though they are together, they will not lose their original identity. Hence, it is called codominance. Next is about multiple alleles. A question may arise in your mind while studying how one allele gets different forms. I think some of you might be knowing the answer. It is because of mutation. It is because of mutation. Variation, remember, variation will bring only the changes, but mutation is responsible to bring the formation of different types of alleles. For example, capital T was the original allele and small t was its mutant. Small t was its mutant form. Capital T was the original allele and small t is its mutated form. That you should remember. So, the best example is for multiple allelism is A, B, O blood group. The best example for this is ABO blood group. And the alleles of A blood group will be IA in homozygous condition, IO in heterozygous condition. The blood group of B, the alleles of B or the genotype of B, IB, IO. Then AB will be IB genotype, only one genotype. Say these two are homozygous condition, these two are heterozygous condition, and O, the allele will be IO. This is homozygous. This is homozygous expression. So now, how many different types of allelic pairs are there for these 4 blood groups? 1, 2, 3, 4, 5, 6. So to express 4 blood groups, how many allelic combinations are there? Total, there are 6 different allelic combinations are there. That you should remember. Then the next aspect is about so what is multiple allele then? Multiple allele means more than two alleles govern the same character. Definition: multiple alleles, more than two alleles govern or control the same character. If more than two alleles, if they govern or control the same character, it is called multiple allelism. Now, fourth one, polygenic inheritance. Polygenic inheritance. What is this? Polygenic inheritance. Inheritance in which some characters are controlled by three or more genes. Inheritance in which some characters are controlled by three or more genes. Three or more genes. For example, human skin color, and human height. Human skin color and human height. See, if one character is controlled by more than one gene, it is called polygenic inheritance. Here, three or more genes, because human skin color, we come across different skin colored human beings. I'll take human skin color as an example here. Human skin color. There are three genes. Assume there are three genes: A, B, and C to control skin color. The dominant form will be A, B, C. Dominant form will be A, B, C. And the recessive form will be small a, small b, small c. The dominant form will be capital A, capital B, capital C. And the recessive form will be small a, small b, small c. Then the genotype of dominant allele. Genotype of dominant allele will be capital A, capital A, capital B, capital B, capital C, capital C. And the person will be having dark skin color, African Negro color. That is a darkest skin color. Phenotype will be darkest skin color. Okay. And the genotype of recessive allele. Genotype of recessive allele will be small a, small a, small b, small b, small c, small c. Small a, small a, small b, small b, small c, small c. And it is the lightest phenotype. Lightest skin color. So one represents the darkest skin color and the other represents the lightest skin color. So in between there are so many type of skin colors that we come across. In between those two colors there are so many different types of colors will come, and that's why varieties of skin colors are there. Now let us see. If the genotype, third combination, capital A, small a, capital B, small b, capital C, small c, then the person will be neither too dark or too light. He will be having intermediate color. Intermediate color. He will be having intermediate color. Neither dark nor white. An intermediate color will be there for the person. So this is about polygenic inheritance. What is polygenic inheritance? One character is controlled by 3 or more genes. The best example is human skin color or human height. I'll take the example of human skin color. Assume that there are 3 genes to represent the human skin color. They are capital A, capital B, capital C. Then dominant form will be capital A, capital B, capital C. Recessive form will be small a, small b, small c. Then the genotype of dominant allele will be capital A, capital A, capital B, capital B, capital C, capital C. Darkest skin color. Genotype of recessive allele will be small a, small a, small b, small b, small c, with the light skin color. And the genotype, if it is with the 3 dominant allele and the 3 recessive allele, then it forms an intermediate phenotype, intermediate color. Now fifth one, we will discuss about the next aspect, that is pleiotropism. Pleiotropism. Single gene exhibit multiple phenotypic expression. If one gene or single gene, if it is able to express multiple phenotypic effect, then it is called pleiotropic gene. Called pleiotropic gene. It is called pleiotropic gene. If one gene, if it controls more than one character, then it is called pleiotropic gene, and the phenomenon is called pleiotropism. The phenomena is called pleiotropism. Okay, dear students. Now, we observe this. We observe this type of genes works in metabolic pathway. Metabolic pathway. This type of gene that works in metabolic pathway. Metabolic pathway means in digestive activity. For example, starch synthesis. Starch synthesis in pea plant. Sickle cell anemia is another example. Sickle cell anemia. Then phenylketonuria is another example. So, dear students, you should remember what is pleiotropism. If single gene is capable to or able to express multiple phenotypic effect. One gene, if it controls many phenotypic characters, it is called pleiotropic gene, and the phenomenon is called pleiotropism. We observe this type of gene works in metabolic pathway. For example, starch synthesis in pea plant, sickle cell anemia, and also phenylketonuria. Phenylketonuria. Next, let us discuss about starch synthesis in pea plant related to the phenylketonuria. Starch synthesis, related to pleiotropism. Starch synthesis in pea plant. Starch synthesis in pea plant. More starch synthesis occurs if the plant is producing or having gene capital B. More starch synthesis occurs if the plant is having capital B allele. Then the small quantity of starch will be produced when the genotype is small b. When the genotype is small b. And when we cross large sized seeds with more starch, its genotype is capital B. Small sized seeds with the less starch, with the genotype small b, then here one gamete will come down, one allele will come down, here one allele. in the F1, we are going to get an intermediate variety or intermediate size. Intermediate size. It will not be neither large nor small. But here, because of this intermediate size, remember dear students, according to Mendel, what it should be? It should be large sized seed. But here, they didn't got a large sized seed, they got intermediate sized seeds. This shows that this example is the example of incomplete dominance.